Welcome, everyone, and thank you for joining us for our presentation titled Simplifying Component Selection in Lateral Flow Assay Development. It is my pleasure to introduce you to Klaus Hochleitner, Global Technology Lead in the Diagnostics of Sativa. For a complete biography of our speaker, please visit the tab at the top of your screen. If you have any questions that might arise during the presentation, we encourage you to submit them in the Q&A box. Our speaker will address your questions following the end of the presentation. Please join me now in welcoming Klaus Hochleitner. I will now turn the presentation over to him. Welcome, sir.
Great. Thank you for the introduction and welcome to this presentation on component selection and lateral flow assay development. Lateral flow assays are rapid tests that are unrivaled in terms to speed and ease of use. The point is that their development can take an awful lot of time. So typically starting from the test idea over component sampling and development and design verification, you can easily spend up to 12 months, and including stability tests, regulatory approval, validation, and scale up, it may last a total of 2 years. You want to get this step correct, and that means that you need to select your components early and in the correct way. In general, a lateral flow test consists of a couple of different materials that serve different purposes. And the sample flows laterally through these pads and membranes and generate your test results. Basically, we discriminate between two assay types. One type is called a sandwich test. That's being used if you can bind two binders to your specific target molecule. Binders can be antibodies, can be antibody fragments, can be affimers or aptamers. The point is that you can bind two of them independently to your target, and they do not interfere with each other. The other option is a competitive test. In that case, you cannot bind more than one binder to your target molecule, and that means you have a test line that already contains your target molecule, and in a negative sample, your antibody, your detector antibody, will bind to that test line, and if the target is present in the sample liquid, you will see a decrease of the signal at the test line, while in the sandwich test, you see an increase in the signal at the test line. Both tests are being used today, and as an example for a sandwich test, that's the well-known pregnancy test, and an example for a competitive test would be a drugs of abuse test. Typically, a lateral flow manufacturing process consists of a number of different steps. So you coat the sample pads and potentially blood separators and conjugate pads with appropriate buffers. You apply your detector conjugate to the conjugate pad and dry it. You dispense your test and control lines onto the membrane. If necessary, you would coat even the wick. You will affix all components to a self-adhesive backing card, cover this card with a translucent cover tape if the test is to be sold as a dipstick, or you will cut the card into strips of defined width and encapsulate these strips in a cassette housing. It's not to be sold as a dipstick. So what we have here is a multi-component system. The advantage of this multi-component system is that this test strip contains all chemicals and reagents that are required, and it contains them in the dry state. In most cases, you need no sample preparation, at least let's say no external sample preparation, because you can add a number of buffers to the sample application pad, which is the first pad in your test, and force your sample to have a specific ion strength or a specific pH. The end user just adds the sample and lets the test run until the results get visible. And that means it can be handled by lay people. The test result is appearing rapidly. A typical test duration time is something between 5 and 15 minutes. The obvious disadvantage is it's a multi-component system and it's very challenging in development and manufacturing. When you develop an LFA, Labeled antibodies or affimers, aptamers, other binders like streptavidin or neutravidin, capture binders, and the test membrane are all integral to the success of a lateral flow assay. And that means you have to have a thorough selection process, and you must ensure that these components work together. And you have to have an eye on the properties of your sample liquid and the specifics of the target molecule that you want to detect. Selection optimization of binders is an important step, but these binders communicate with the membrane, if you want to say so, and that's sometimes not being addressed correctly during LFA development. Let's talk about that reaction membrane. So the key considerations is on a couple of parameters which we will discuss as far as they are important for your test development. And we will talk about capillary rise time, about membrane backing, about blocking the membrane, and about membrane surfactants. And we will come back to that specific point later. Very often, the correct choice of the membrane will come down to the nature of the test being designed, and it may be reasonable to test many product variants. So capillary rise time, what is that? That's the time that a liquid, and typically this is water, needs to flow a defined distance on the membrane. And typically this is 40 millimeters, and that's being used as in-process controls in membrane manufacturing, and it's what we call a specification parameter. And at the lower left of that slide, you see what determination of capillary flow time or capillary rise time looks like. Typically, membranes are characterized by a specification that has a specification mean, for example, 80 to 100 seconds for a very fast membrane. And it has specification limits, which we'll discuss in a minute. Very fast membranes are being used for things like saliva, resolubilized solids. Medium fast membranes, which typically have a flow time of approximately 120 to 135 seconds as specification means, will be used for things like blood, serum, and plasma, or for urine. And very slow membranes, which have a flow time of roughly 180 seconds, will be used for things like water. All suppliers manufactures the membrane to a defined capillary rise time. And that's the main specification parameter when we manufacture membranes. While this capillary rise time specification has an upper and a lower limit. And that means you have to take care that your reagents or your test results are working the same way on both ends of the specification limit. If you have an issue with membrane specification parts at the fast end, you can easily see false negative results. The sample runs too quickly across the membrane, and you do not see the result on the test line that you would like to see at the detection limit. If you have an issue with the slow part of a membrane specification, you may get false positive signals, or you will have an issue with membrane clearance. That means the membrane is not white again when the test duration time is over. In rare cases, we see issues at both edges of the specification, and then it's getting really difficult. What you should do is you should test membrane samples from both edges of the specifications, something that we call limiting samples, and look for the performance of your test. It might be that you have to select a different membrane grade if you run into trouble, or it might be that you will have to ask for a customized membrane, a membrane that works with your test and has its own capillary rise time specifications. An important point here is that binders, although they have the same affinity constant, may be different in terms of the on and off rate of the binding reaction. For lateral flow tests, you would be interested in very fast on rates because the interaction times that you have available at the test line, but also for the conjugate, are very, very short. Antibodies that have the same affinity but have slow on rates and slow off rates are typically ELISA antibodies. They will not work perfectly well in lateral flow assay tests. So if you have a bunch of antibodies available, you should try to get surface plus one resonance data that describes the on and the off rate of the binders. If you have just one pair available and no choice for selecting another reagent, you will have to play with the speed of the membrane. So if you have to use binders that have relatively slow on rate, you will have to use a slow membrane if you want to have a chance to reach your detection limit. Membrane backing. Well, what is that? When you manufacture nitrocellulose membranes, what is being done is the nitrocellulose raw materials are dissolved in organic solvents with boiling points lower than that of water. These casting mixes are poured onto a solid surface, typically a steel belt if you want to have an unbacked membrane, or a plastic belt if you want to have a backed membrane. The membrane is formed on that support, and in case of a backed membrane, it's not lifted off the support, so it stays there. The advantage is its mechanical stability. You will not be able to tear that apart. The disadvantage is you have to live with the upper side of the membrane. And what that means, you see on the right part of that slide. What you see here is two electron micrographs of an unbacked membrane that's called A100. That's a fast lateral flow membrane. And what you see that the upper side, which we call the air side, has a relatively coarse structure, obviously a random structure, which is typical for nitrocellulose membranes. But the right side, which is the belt side, the part of the membrane that was forming directly on top of this support steel belt, has a denser structure, which means a higher protein binding capacity, and it looks more regular. So it's not as variable as the air side. If you want to use an unbacked membrane, use both sides, and it could be very advantageous to use then the belt side. The downside is the mechanical stability of an unbacked membrane. And especially for beginners in the field, our recommendation is use the backed membrane. The nitrocellulose matrix of the membrane binds proteins, and you like that. You want this membrane to bind proteins as long as you dispense your tests and control lines. As soon as these binders have been bound by the membrane, you are left with an awful lot of membrane area that's still open, has not bind anything yet. This membrane would bind anything it would find in your sample liquid, your target, your detector reagent, whatever you offer. And that means you will have to block these free areas in order to prevent them from interact with your target or your detector. And there's two options. At first, you can do something that's called block on the fly. In that case, blocking reagents are part of the pre-treatment buffers of your sample pad or your conjugate pad. These reagents are dissolved by the sample liquid. They migrate with the sample and block the membrane while the sample liquid is moving across the membrane. And that's the reason why it's called block on the fly. The advantage of this process is a cost reduction, because the alternative is blocking in a separate manufacturing step. In that case, the membrane is treated with the test and control line reagents. The reagents are dried, and then the membrane is soaked in a buffer, or you spray the buffer onto the membrane that contains the blocking reagents. A separate manufacturing step means another step that needs to be tested, developed, validated, and of course it's the cost factor in manufacturing. The advantage is you have full control over the amount of blocking reagents that you apply to the membrane. If you do block on the fly, you have to live with the thickness variability of the pads that are in front of the membrane, and that means you will have different concentrations of blocking reagents in your migrating liquid, since you will have variable thickness, and that means variable pad volume, but you will always apply the same volume to your cassette housing or to your dipstick. And that means your sample liquid is always the same, it's always the same volume, but the amount of molecules dissolved in it when it runs through the pads will differ. That's especially difficult when you want to develop a quantitative test system. And for quantitative tests, a separate blocking step of the membrane is more or less unavoidable. Many people are not aware of the fact that the reaction membrane contains a surfactant. All nitrocellulose membranes contain surfactants. And the surfactants are needed because the nitrocellulose membranes are quite hydrophobic matrices, and only the surfactants make them really hydrophilic. So surfactants have been a part in nitrocellulose membranes since their very infancy. And the oldest patent that I could find was from 1917, so it's more than 100 years old, and even that membrane already contained the surfactant. In protein applications, not only lateral flow, but also flow-through tests or Western blots, these surfactants help to immobilize your binding proteins to the membrane matrix. Different suppliers use different surfactants and apply them at different stages of the manufacturing process. So it makes sense to test membranes from different suppliers for your specific application. This denaturation process binds proteins to membranes, and you need that. The problem is this process denatures proteins. It can easily be that in the monoclonal antibody or the affimer or the aptamer that you are using, the surfactant destroys the binding site, or at least reduces its activity. In that case, you may have to switch to another membrane that contains a different surfactant, or worst case, you may have to look for different binders. I have worked with more than 1,000 different monoclonal antibodies and all kinds of other binders during my work in lateral flow diagnostics. And from what I've seen is 2 to 3% of all monoclonal antibodies, that means 2 to 3% of the clones, do not work on membranes because the antigen binding sites are destroyed by anionic surfactants. And in that case, although it's a rare case, there is no way out. You can't make that run on a nitrocellulose membrane, and you have to look for a different binder. But even if you can immobilize your antibody successfully, you should keep in mind that this immobilization is a random one. That means the antibody is not bound with its Fc part to the membrane, and the antigen binding sites are protruding into the free migrating liquid. Any orientation of the antibody is possible, and there's two things that you have to look for. One is that a preferential orientation that blocks the binding sites because the binding sites binding to the membrane? In that case, you have a problem with the antibody-antigen interaction. And the second question is, how close are the two binders to each other once they bind to your target? If they are too close, you will run into trouble with steric hindrance, especially in membranes. That can easily happen. And our recommendation is you should do epitope mapping using surface plasmon resonance to make sure that your antibodies are doing what they want them to do without interfering with each other. It can also give you reliable data about on and off rates, and you could also test for inconsistencies from batch to batch, and you can test for lack of compatibility with the use of specific reagents. What you also have is pads on the left and the right side of the membrane. So you have a sample pad that you can coat with buffers that may contain reagents that block the membrane, but typically, and much more importantly, they contain reagents that take care that at least some key parameters of your sample will always look the same after the sample has dissolved this buffer that you have coated into the sample pad. An example is urine. Urine is coming with a quite acidic pH in the morning, and it gets alkaline in the same patient towards the evening. So a typical range in pH is something between 6.5 and 8. What you can easily see that especially specific binders like monoclonal antibodies change their binding characteristic when you change the pH. It makes an awful lot of sense to take care that every urine, regardless when it was collected, has the same pH when it runs across the test. And this is what you achieve in the sample pad. Your sample pad may also be a blood separator. As the name says, it's being used when you use whole blood as a sample, and its task is to remove the blood cells, especially the red blood cells. Still, the main detector particle in lateral flow rapid test is gold nanoparticles, and that's red. And if your red blood cells leak onto your membrane, you try to look for a red signal on a red background, and it's not very likely to give you very good signal-to-noise ratios, and that's why you need a blood separator. And then you have your conjugate pad. That's the most tricky part of the test, to be honest, because what you expect it to do is you expect it to absorb the conjugate pad that you dispense on it in the liquid form, and you definitely should dispense and should not soak the pad with the detector. Then you dry the conjugate in the pad, and you would like this conjugate to remain stable and not be destroyed during the drying process. And then you expect, after a year of storage in a drugstore, a household, a doctor's office, under more or less dubious environmental conditions, you in earnest expect the conjugate pad, when the sample enters the conjugate pad, to release this dry conjugate into the sample and make it run across the test. And the detector reagent on the conjugate is supposed to find its target, to bind it as it would do it before you have applied it to the pad. And that's a very interesting expectation. And mistakes during preparation of conjugate pads are the most likely root cause if you run into trouble with your rapid test. And finally, you'll have an absorption pad, often referred to as a wick. The task of this pad is to capture a sample liquid and to retain it. What you should take care of is that the absorption capacity of this wick is much larger than the volume of the sample liquid that you apply, because what you have here is a matrix that generates your results, that's the membrane, which is very thin compared to all other pads. It will dry first, and then you create a concentration gradient, and what you can see then is a backflow from the wick onto the membrane, which will give you all kinds of unspecific weird phenomena on the membrane, and you do not want that to happen. You would like to have the membrane wide as long as possible. Some FAQs and some common issues. Very often I'm asked, what is the pore size of your nitrocellulose membrane? In plastic back membranes, the pore size cannot be determined directly. That's due to the plastic backing. And that's the reason why all membrane manufacturers refer to capillary rise time or capillary flow time as a specification parameter. As a rule of thumb, a very fast membrane has a very large pore size. And a given sample liquid will run faster through a very fast membrane than it does through a slow membrane. The point is, depending on the properties of your reagents, especially of the capture reagent on the test line, a fast membrane is likely to give you a lower sensitivity as compared to a slow membrane. As said, it depends also on the properties of your capture reagents. A capture reagent with an extremely high on rate gives you a very good sensitivity even on a very fast membrane. A capture reagent that has a disaster of an on rate might not even work on the slowest membrane that you can buy in the marketplace. Another question is, what adhesive is used in your back membranes? No membrane manufacturer is using an adhesive to create a plastic back membrane. These back membranes are 100 micrometer thick polyester supports onto which the casting mix is poured, and the membrane is formed on this polyester support, and it stays there by simple adhesion. Adhesives come into play once you laminate these membranes onto the backing, onto which you also have to affix all the pads that you need to create your rapid test. A couple of issues is listed on the right of the slides, including possible solution. So what is frequently seen uneven lines or dots during the dispensing process. You may use different membranes with different capillary flow times. You may use the dispensing rate of the reagent, perhaps including reducing the dispensing volume. If you do that, you may have to increase the protein concentration of the reagent. You will take care of the buffer composition, and you will check the dispensing process. And although many people don't like to hear that, in a sizable number of cases, the reason for uneven lines and dots is lack of maintenance of the dispensers. So it makes sense to clean them properly and to take care of their function. False positive signals in terms of membrane issues can be modified by changing the buffer in the conjugate. You may tweak the pH, the salt concentration. You may play around with surfactants, and worst case if that you have to change a conjugated protein. For false negative results, a typical solution would be to reduce the capillary rise time. That means to use a slower membrane. This very often leads to a better sensitivity of a test. Increasing the sample volume is also an option, but there are limits to that in a lateral flow test. So the typical sample volume is 100 microliters. You can use something like 2, perhaps 300 microliters. The idea to run 1 milliliter across a lateral flow test is simply not feasible, so there is an upper limit to that. What is also sometimes seen is uneven liquid fronts of migrating samples. It might be that you have stored your membrane too long, but in most cases, it's a problem with either that you're using a relatively hydrophobic membrane. That means a membrane that has a very low surfactant content. And the other option is that you have a problem with the relative humidity in the room. And if this is too low, then you will see flow issues. The question with the hydrophobicity of membranes is a very interesting one. Hydrophobic membranes, or let's say membranes that have a low surfactant content, they're not really hydrophobic, can easily give you better sensitivities because they have a less negative impact on your binders, especially on the test line. You can modify the hydrophilicity of the membrane then by playing around with the blocking buffer. So a membrane that is relatively hydrophobic, and you like that because it gives you a better sensitivity, should be blocked in a separate manufacturing step, because then you can deal with any uneven liquid fronts or migration issues that you see and still have a better sensitivity than you would have with a high surfactant membrane. So to summarize that, the nitrocellulose membrane is the core of your lateral flow assay because this is where the end user sees the test result. Getting the right membrane first time, or at least as soon as possible, is key in keeping your lateral flow test development and on track within specification and on budget. Choosing a membrane is not just simply using a paper. It needs to be considered alongside a lot of other key decisions, and it has to be done in conjunction with reagent selection. The membrane needs to be able to immobilize antibodies. It should not affect their binding capabilities, and it must enable the sample to flow at the optimal speed. And it makes a lot of sense to work closely with your membrane manufacturer to discuss common pitfalls, to discuss what can be done and what cannot be done with a given set of reagents, and perhaps to design in a membrane that's manufactured according to your needs. And if you have questions, please feel free to talk to us. Thank you.
Thank you, Klaus, for that outstanding presentation. We will now move into the live Q&A portion of the presentation. Klaus will unfortunately not be able to join us for this portion, but we do have Lee Jenkins joining us, Senior Product Leader for Sativa. Welcome, Lee. And I just want to remind our audience members to please submit their questions via the Q&A box. Looks like we already have some great questions coming in, Lee. So let's get started. Our first question is, what are the common reasons why people fail to develop a viable test?
Okay, yes, thank you. Yeah, just to repeat, apologies to everybody that Klaus can't be here at the Q&A session. But hopefully I can help answer these questions, and anything you wish to discuss with Klaus, we can take offline. So with regards to the common reasons why people fail, Klaus just did an excellent job. In the video, I was talking about different reasons, but essentially, I think to summarize what he presented, there are many different factors involved in developing the test. So it could be anything from the detection pair, your detection reagents, reagent used, which pads, membranes you select, the backing card selections, or even the working environment and the equipment setup. So every single factor involved will affect the overall performance of the test. So in terms of test development failure, it really does depend case by case. And we can help you with your troubleshooting if you need this support. But we support that. We suggest that look at maybe the capture reagent selection or the texture selection and reagents of the tests are probably a key place to start. But unfortunately, you'd have to test all of those items.
Thank you so much, Lee. Our next question, why does a test take so long to develop? For example, the COVID-19 test.
Golden question. So everybody wants the quicker development, and to develop a normal lateral flow test can take 2 to 3 years typically for a customer to get from development, get through clinical trials, and get onto the market. To develop a good, robust test, normally you take an absolute minimum of 1 year at the R&D phase. So to go through the whole tech transfer and scale up after that, the stability trials obviously requires time as well. So everyone's working with absolute urgency during the current pandemic to get tests as quick as possible. But we need to allow time for that development, for the optimization, in order to fulfill all the needs. Understanding COVID is a special situation, and we do have some emergency use authorization to bypass some of the timelines. But it still takes time to go for such a complex development. And hopefully Klaus's 30 minutes this presentation touched on some of the elements of why it does take quite so long to complete the process.
Thank you so much, Lee. Can you talk to the audience a little bit about the development of challenges of multiplexing?
Okay, yes, another good question. So I guess first of all, there's several different types of multiplexing assays. So you could have an array of strips. You could have spatial separation of detection sites. You could have a broad selective recognition element, or you could have a variable signal reporter. And the primary consideration when contemplating multiplexing in a lateral flow assay would be about the cross-reactivity and interference between the assays, because this might impact your signal. The sample treatment method compatibility, the sensitivity and dynamic range of each of the parts of the test and each of the targets that you're trying to detect, the stability, and the assay format. If there's a mix of effective and sandwich assays involved, then this has a whole extra level of complexity. When talking about a multiplex assay, a membrane which nitrocellulose you select is a key element. So we suggest that when you develop a multiplex flow assay, they need to consider upfront the number of test lines they require, the viscosity of the sample, the desired or at the very least acceptable test run time, the kinetic properties of the capture reagents, and then, as Klaus touched on in this, the membrane surfactants obviously very important, and that would need testing and sampling of different membranes to understand what works well with your assay.
Thank you so much. And I want to thank our audience members for these great questions coming in. Lee, what blocking agents are typically used, and how does this impact flow rate, for example?
Okay, so normally we block our conjugate pads and nitrocellulose membranes. For the conjugate pads, the purpose of the blocking is to make the conjugate stable during the whole shelf life of the test. So make your conjugate release efficiency and repeatable from the conjugate pads. You can start to block your conjugate pads by developing several different formations. So we have a standard process of a borate, a BSA buffer that we use, but I'd suggest that if you want to get in contact with our teams after the meeting, we can help share some details of what we've used previously. But people need to keep in mind that different part materials and different tests will require different buffers. So there's not one bespoke solution, which is why I'm a little bit hesitant to share too much on this question. With regards to the nitrocellulose, the purpose of blocking the membrane is to reduce the unspecific binding of the protein. So different concentrations of BSA can be tested as a blocking buffer here to see how the impact is on the overall test.
Thank you so much. And how do you start to evaluate membranes based on what surfactant they use?
Okay, so yeah, I think Klaus did, again, a session on this, but the surfactant is a very important factor of a nitrocellulose. He mentioned that all nitrocelluloses on the market have surfactants present, generally different types. So you need to understand that not all monoclonal antibodies will work well in a presence of a given surfactant. That antibody testing in the presence of various surfactants is critical to optimize the performance. So we've got a feeling our side that about 3% of monoclonal antibodies will not work in the presence of any surfactants. So others may only work at low surfactant concentrations. So it's again very much around that testing, evaluating, trying to test as broad a range as possible during your early development stages. Different surfactants in the membranes may lead to different test results. So you may get changes in sensitivity, positive or negative influences on your test, or you may get false positives if you are not compatible. So that's really key to the testing element. And then also the way the surfactants introduce the membrane can have a big influence on the results. So we have different product families at Sativa nitrocellulose where the majority of our grades, such as the FFHP grades, we add surfactants, the cast-in mix. And we do this because we believe that it gives a more consistent mix of the surfactants to the membrane. Some other product families that we release or that our competitors have available, they post-treat the product after manufacture with surfactants. Again, it can have a different impact on your performance, positive or negative. So we encourage you to test both to see what works well for yourself. But it may lead to more variability or less consistency of surfactants if you use a post-treated membrane. The type of surfactants, concentration of the surfactants, they're all proprietary to each membrane supplier. So they're not going to necessarily tell you what surfactants included, but we suggest if you talk to our application specialists, they can give you more information on what they regarding our membrane surfactants and what could work for your test.
Wonderful. Thank you so much. And Lee, what are the scale-up factors that one needs to consider during the test development?
So before I answer this, I guess the best answer for today is there's a session later in the summit about scaling up your development. So we got Aberdeen Health talking about the importance of a contract manufacturing organization and talking to them early in your development. So join John's session later today to understand exactly what's involved there. We also have Biodot coming on at the end of the event to talk about how they can bring scale to the test. But from our side as a component supplier, we suggest that you choose materials from high quality suppliers with appropriate levels of technical support. Ensure the membranes you are purchasing are available at a large scale and that there's no capacity concerns. That's especially critical at the moment because around COVID-19 we're seeing some suppliers are having a run on membranes that have the normal level of availability that they would have had previously. Consider dispensing membrane reagents using non-contact methods. So this can help your automation and the volume possible. Avoid immersion processes where possible. Use covalent conjugates, which are appropriately optimized and stabilized. What other suggestions have I got? Use inline processing and automated process controls. Use a controlled manufacturing environment. And then in your development, concentrate on appropriate device design and assembly methods so that you've got a test which can go from a lab-based test into a full automation scale test. Again, if anyone needs any support over these scale-up conversations, please speak to our teams. They can help guide you on suggestions of what we can provide here.
Thank you so much, Lee. And thank you again to our audience. This next audience member poses a question from their own lab and hopefully can get some assistance from you. He writes, besides we consider the capillary flow of the membrane, is there any other factor which could help to control the capillary speed? For example, they have a problem with their capillary flow right now in that their assay is too fast and they've tried to add surfactants or even blockers for the membrane, but it wasn't significant. Can you help them with this problem?
I can, but also I suggest that if they want to reach out to any of our contacts or any of our field application support teams, they can discuss very specifically. But at a high level, the advice I can give is that the surfactant generally will not help slow down capillary flow time. This is not something that the surfactant brings to the membrane. Blocking can help a little bit, but again, not a huge contribution. The most significant way of affecting the flow rate would be looking at the membrane and the pads that you use for the assay. So the wick and the sample conjugate pads, what is the wicking rate of those, but also what's the capillary flow time of the nitrocellulose. So try pads and membranes which have a slower rating for the wicking and capillary flow times. So all membranes and pads are released with defined membrane speeds. So talk to your supplier, understand what they have, what membranes you have now, and where that is in the range, and try to select something with a higher reading, so a higher measurement, which gives you a slower membrane. You can even talk to your supplier and see if you seem to fit in between two pads, you're not getting a correct level of sensitivity, to see if they can give you a custom flow membrane, which is at the higher end of the specification to the test. Outside of that, it's probably looking at the affinity binding rate of your detection pair. You need to go back to do something like SPR analysis to understand the on-off rates using Biocor or something similar. But this can often mean restarting the whole development process. And we would suggest looking at the pads in the membrane, your first simpler step.
Wonderful. Thank you so much. I hope that helps her. Our next question, can the choice of antibody type, for example, antibodies, aptamers, affirmers, affect the flexibility of choice of NC membranes and ultimately the speed of the assay?
Yeah, another one where it really depends. So typically the capture reagents will work differently with different capillary flow times and surfactant types. I wouldn't say that there is any restriction or flexibility around choice, but it goes back to the mantra doing development: test as many different varieties as possible when you're selecting your membranes and your pads. Test different families, different speeds, just to understand what performance you're getting. If you're able to test your affinity binding of the pair via the SPR analysis, which I just mentioned, Biocor is a way of doing that, then again, it can help you understand what capability you have within your test. But unfortunately, it's going to be a trial and error activity for the customer.
Thank you so much. We have time for a few more questions. Is it possible to obtain a highly sensitive and quick reaction by changing the antibody type rather than the membrane itself?
Yeah. Okay. So if you have a test which is running at present with a antibody and you're looking just to switch that over to aptamer or aftermer, that's highly unlikely because it's very rare in lateral flow, and we test that you are able to make a simple swap out of one component to another, especially something as critical as the detection reagents. You probably would have to go back through the whole redevelopment, and essentially it'd be close to developing a new test. Looking at different detection types, you could get faster speeds, and you might need a more sensitive assay, but it all really depends on your affinity binding rate and how it's compatible from all components in your test. So again, not a direct answer, but it's very specific. If you have a test that you want us to help you talk to, again, I encourage you to contact one of our team offline and we can help discuss this with you. But there's not a one-size-fits-all solution to this answer, unfortunately.
Thank you so much, Lee. Now, if we inject, for example, 2 proteins into a membrane, but when checked only one protein appears, what does one do? And whether do they replace the membrane or the process for the protein that must be correct?
Yeah, it's awkward to discuss very specific cases such as this in a general format, but know in more detail. I don't want to keep referring to the same response, but if the customer is able to contact us, we can set an NDA and talk a lot more detail about their test and understand exactly what they're doing to consider all aspects. From a high level, it's quite difficult to say what could be the unfortunately.
Thank you so much.
Yeah, unfortunately, that's one we'll have to defer to offline today's call.
No problem. How do you minimize serum interference in the serology LFA?
Okay, so the simplest way of minimizing interference is by having a good blood separator. Klaus touched on in the presentation very briefly, there's different blood separators available, but you should essentially try different sample volumes and different blood separators to understand what will best for your test. There is a lot of information out there, and different suppliers who have different separators. If you need more advice, I advise person to look through to the Cytiva website, where we do have some content around our different blood separators and how this can work within their test. So encourage you to get in contact if you need samples from us to see what works well, and we can happily supply these to you.
Thank you so much. Now, Lee, our next audience member has a question about validity and reliability. If things like humidity and the shelf life of the membrane can impact the test, and assuming packaging can mitigate this to an extent, but not completely, would the typical validation line at the end of the test be the only method to determine a faulty test? And if so, how reliable will it be?
Interesting. So the validation line, I assume you mean the control line. So yes, this would be, obviously your North Star, as we say. So this would be your ultimate way of understanding once your test is developed, if it's failed. But I would go back to that this is all about development, this question. So ensuring that you have a rigorous development, that you are fully testing the stability during your early processes, that you are understanding the capability of your assay. You don't want to do this once you've got through development and you've got a product on the market. If you're relying on a control line to tell you whether a test is faulty or not, you're in serious trouble. You want to understand all of this very early in the process, understand the shelf life of your conjugates, how stable they are once they spend someone's stride, understand the stability of your whole test. And the only way to do this is by stability trials and a rigorous development process.
Thank you so much. And again, what a great, lively conversation from our audience today. What are the reasons for getting ghost bands with capture antibody on the test line, and how do I get rid of it?
Again, it's one that sounds very specific to this customer's assay, so hard to give an answer which is going to be definitive, providing a solution to them. So again, feel free to contact our teams offline where we can help discuss this. But Klaus outlined in the presentation some ways where we have, faint lines or these ghost bands. It's all generally about looking at your non-specific binding or having a look at your dispensing rates around the test. It's something which, we could help talk more details offline, but it's very difficult again in this forum to kind of give you a definitive answer on what would work for this test, unfortunately.
Wonderful, thank you. And the same audience member wants to know, what is the optimum capture antibody concentration for the test line?
Yeah, again, it's going to be very specific to your test, what antibodies you're using, what you're trying to detect. Not able to give you a definitive answer on that in this forum. If we want to talk about, we can give you some examples of what concentrations we use for different targets. We can say from our experience what's worked well, but such a broad statement across the whole vessel flow, very difficult to comment, unfortunately.
We have time for one more question. Lee, I have a problem. This is coming from an audience member. I have a problem with assay sensitivity and suggested to add stacking pads on it. Could you recommend some suitable properties for the stacking pad? And should we treat the stacking pad with a buffer, for example?
Yeah, so if you've got sensitivity issues in your test, then this could help, but by no means it's going to be the only solution for you. Difficult, again, to understand without knowing what your test is. Important considerations when looking at your pad would be what wicking rates you want or what absorbency you want from your pad. You might want a buffer, whether that's a chase buffer or whether you want to add a blocking solution. They could help. But again, we really would need to understand your specific assay in order to comment in more detail over what would be appropriate for this test, unfortunately.
Thank you so much, Lee Jenkins. We are out of time. I want to thank you. Would you like to provide any closing remarks to our audience before we close today?
Yes, just thank you to everybody who joined in. I've mentioned quite a few times that there's not a one-size-fits-all answer for everything, and that it is very case-specific. So, please, I encourage all of our customers, if they are interested, we are more than happy to continue this conversation offline. Get in contact with your local account managers or your local field application support, and we can help talk more specific to your test and what we can do for you. Thanks, Susie.
Thank you again. Thank you again, sir. And I want to thank Klaus for his time as well. And I want to thank our audience for their outstanding questions and their participation. Just as a reminder, any questions that were submitted but not answered today by our speaker will be addressed via email. And this presentation will be available for on-demand viewing for 12 months. Please remember to share it with your colleagues who may have been interested in this topic but missed today. And don't miss out on the other presentations on our agenda. Visit the agenda tab in the auditorium for a full listing. Take care, and thank you again for your participation. Until next time, stay safe and stay healthy. Bye-bye.